All right, good morning. My name is Gabriel Downs. The time is currently 8:40, and I'm going to talk to you guys about how does the SARS-CoV-2 vaccine work and pretty much give an insight on the structures and aspects that are taken into account when developing the vaccine. All right, so just some basic COVID-19 structures. So we know it is a positive single-stranded RNA genomic structure, and there are four main proteins to take into account. We have the S protein, the M protein, the E protein, and the N protein. The S protein, which is going to be the most crucial in this situation, is the outermost antigen and is responsible for directly impacting the host cell as it is the main form of attachment. This includes the S1 and S2 subunits, which we're going to talk a little bit about later on. And this protein attaches to our ACE2 enzyme, which has direct correlation to our respiratory system. Next, we have the membrane protein. This creates the shape of the viral envelope as it interacts frequently with other proteins along the nucleocapsid. Now we have the E protein, the envelope protein. This is very, very essential and important for the assembly and the releasing of the newly constructed virus. Additionally, it is efficient at organizing the intracellular activities during viral replication. This is also worth noting that it has direct ties to the Golgi apparatus and the endoplasmic reticulum. And lastly, we have the N protein, the nucleocapsid protein. This is what halts the host cell defenses, features as a suppressant, and allows the RNA from the virus's capsid to begin replication. So towards the right, we have a pretty basic coronavirus representation. And we can see that the S protein is protruding out of the cell membrane, which really is an incredible feature because this is what allows it to bind to our ACE2 enzyme, as mentioned before. The second most common protein that along the nucleocapsid are the M proteins. As you can see, they are littered all around the entire genomics or structure. Then we have the E protein, which of course is very, very helpful for the viruses in the releasing of the genomic information, I guess we could say. And then we have the M protein, which is attached to the viral genome, the RNA viral genome. And this, of course, is going to act as a suppressant, as mentioned before again, and have implementations on the host responses. Okay, so how does this basic information impact the vaccination process? Firstly, how do researchers develop the foundation of vaccine? Well, this is done by genomic sequencing. Genomic sequencing can be DNA or RNA, and this is used for bacterial or viral infections. So, as we may know or may not know, vaccines are created by using dead or partially weakened bacteria. You can also do live attenuated vaccines, but this won't be correlated to what I'm talking about today. So the template is created by comparing and contrasting the newly discovered strain to other strains to figure out the background. So to give a better insight on this, when SARS-CoV-2 first came about in Wuhan, China, they automatically didn't assume it was a coronavirus. That's you know you can have hints because you can say, oh, it's a respiratory uh, virus, so it might have ties with the coronavirus. That's a good educated guess. But to make sure, you have to run the sequencing. And what they found is that it had strong ties to the coronavirus family, hence, it is a coronavirus. It's just a new one, it's a new, well adapted one. And lastly, the SARS CoV 2 vaccine is implemented by targeting the S proteins. As mentioned before, this attacks the S proteins, but it goes to the S2 subunit and the S1 subunit, I guess. All right, so speaking of S proteins, this is a good diagram and GIF that allows you to see how the S proteins protrude off of the cell or the virus's envelope, and this makes it easy for it to bind to the ACE2 enzyme, which, as mentioned before, has direct ties to our respiratory system, especially our lungs. Okay, so let's apply this information. There are two main approaches when developing a vaccine. You have the basic RNA slash mRNA approach, which, of course, RNA virus is going to be correlated to mRNA vaccine. But this is done by having the mRNA sampled from the viral strain. So it's actually sampled from the coronavirus and then ejected into the patient. Now, this may sound scary, but this is not what the issue, this is not going to create any issues theoretically. The mRNA is the coding for the S protein of the coronavirus. So essentially, as mentioned in this next bullet point, the body is able to code the mRNA sample to create antibodies. That's the approach. And so once invasion is detected within the body, antibodies will be created to bind to the S proteins, inhibiting further damage. So as we can see in the image to the bottom right, we have these Y-shaped structures, which are going to be our antibodies, just a diagram of it. And they are going to bind to the S protein. So the coronavirus particle will not be able to bind to our ACE2 enzyme. So essentially, it's neutralizing it. It may still be there, but it's neutralized. Now, as mentioned before, we have the S1 and S2 subunits. These are of course impacted, and we have the RBD that is used for the ACE2 receptor. The RBD plays pretty crucial role within the binding and contact. Okay, lastly, we have the DNA and RNA approach, or I should say mRNA approach, excuse me. So basically within this one, a portion of the viral strain is segmented off and implemented into the plasmid. So a plasmid is a circular genomic structure. So you're essentially taking a slice of our plasmid and taking that out while then putting the new portion of the viral strand into it so it can bind and code essentially. And the plasmid vector will interact and suppress the protein expressions, therefore inhibiting them. Now, the DNA approach also targets the RBD S1 subunit, while on the other hand, this was very interesting for me because I wasn't aware of this, but the pDNA, plasmid DNA, vaccines are more durable than the regular RNA. So from reading and researching this, the plasmid DNA is actually easier to transport and get to the patient. So I would assume it has a stronger envelope, possibly. And yeah, I just thought that was very interesting. So going on to our last bullet point, we have optimal dosage in early stages were unclear. Of course, we initially heard that you only need 2 shots. Then a couple months later, there was I can't remember his name, but there was a scientist that came out and said, "Oh, we might need 3 shots." Of course, the public did not take this lightly, I should say. And now we're just back at 2 shots. But you can get a third shot, which is just a booster shot, which is very good because it boosts immunity. So either way, you're doing a favor to the public by getting this vaccine. And this is basically the whole rundown on how the vaccine is created and implemented into the body. And as we know, or if you may or may not know, the new vaccines, I should say, are all DNA, mRNA approached. So that was fun to know. And these are my citations. And yeah, I'm all done. Thank you so much.